All right, well, it is a pleasure to be here. I will leave it to history to decide whether or not NGLY1 is truly a CDG. As Lynn explained this morning, it is in fact perhaps a new kind of CDG. And I'll explain a little bit of the science to see if you believe that or not. But what I'm really here to do today is explain the actions of our group, which is what the title of the talk implies. And I think this is quite appropriate because I've actually been on like something of a campaign lately, a campaign against this phrase, the phrase not actionable. I think it is long past time for clinicians to stop saying it. I think it's long past time for patients to stop believing it. And I'll try to illustrate this to you today by showing what we in the NGLY1 community have done. So it really is time to relegate this phrase to the dustbin of history. And in the interest of time, I'll go very briefly over the history of NGLY1, which somewhat necessarily has to begin with my son Bertrand. So my son Bertrand is actually the first patient ever discovered with NGLY1 deficiency. And that all came about because we began our journey, as many of you have done, on that undiagnosed island that Ellen mentioned. So we all know what it's like to be on this island. We all know what it's like to watch our children suffer and to not know why. It's a terrible place to be, but we've all been there. So in many ways, this is our story. But it's also an island where I think many of us learn about the strength and resilience that lies inside the human spirit. And certainly we learned that as well. So that was our life for 48 months. That's where we were stuck, stranded, until a team of scientists from Duke University, who we'd been working with for quite some time, said, we think we have a way to diagnose your child. So they proposed to do trio sequencing on my wife, my son, and in me to look for mutations that could be causative of his condition. And what they found was he had two mutations that he inherited from both my wife and I in his NGLY1 gene. So they weren't de novo like suspected, they were in fact inherited. And then things got a little bit strange, because when they said that he had these mutations, they also said that he is the first patient we've ever seen with loss of function in this particular gene. He's literally the only patient out there in the medical literature. So we're not even totally sure that this is the cause, but we're pretty sure that this is it. So we had some homework to do. So the Duke team kept going. They checked to make sure that there was in fact no NGLY1 protein, and there wasn't. But that's not definitive yet. What we really had to do was try to take the phenotype of my son, all the data we'd collected over the years, and map it back to this purported genotype, this loss of function in NGLY1. And this requires actually asking, what does NGLY1 do? So what does NGLY1 do? Well, NGLY1 is a gene that encodes a protein called N-glycanase, and N-glycanase is responsible for deglycosylating misfolded glycoproteins after they're retrotranslocated from the endoplasmic reticulum. So what this causes in the absence of NGLY1 is presumably an accumulation of these misfolded glycoproteins in the cytoplasm. And there were clinical indications that this is exactly what was happening, particularly in tissues like my son's liver. So they said, well, this is better evidence, but how do we really know that this is truly a novel CDG? Well, there's really only one way to know that as far as I can tell, and that's to ask Hud Freeze. So they turned to Hud and they said, Hud, here's the data. If you saw a patient like this, what do you think you've got here? He said, what I think you've got is a new CDG. So that's how NGLY1 came into being, or how I mean, it's been around for a long time, but that's how we realized it was there. And at the same time we got this diagnosis, we also got word from the clinical team at Duke that this was not an actionable diagnosis. There's literally nothing you can do because it's a brand new disease. There's obviously no medications. So, geez, that sucks. Well, I really don't believe in this phrase. I think it just doesn't belong in our medical vocabulary. So let's talk about what happens when you get a not actionable diagnosis like NGLY1. Well, for starters, as soon as you have a name, you have, instead of dozens of potential targets, all those diseases you thought your kid might have, you've got just one to focus on. So we started focusing on just the one. And as soon as you have a gene name, and as soon as you have something you can screen for, you can actually start doing science. And I think in this case, science is what becomes the medicine. So even if there's no medication you can take, you can do science. So science in many ways becomes that action. And so let's talk about some of the actions we've taken in the NGLY1 community over the past 3 years. Well, for starters, my wife and I could actually have another child safely. So maybe some clinicians would consider him not actionable. But I guarantee you, if you've seen him running around, he's highly actionable. Way too much action. And we headed, I think, within weeks out to Hudfreeze's lab, and we said, well, can you just tell us where we are? Where are we in this landscape of glycobiology? Where have we arrived? So he really took us to the peak of our diagnostic journey and showed us how far we'd come, and ultimately how far we would have to go, and in fact, how far all of us have to go. He said, if you want to reach understanding of treatment and cure, you have to summit Mount Glyco. That's the mountain all of us in this room have to climb when we're fighting a CDG. And it's not easy, but as parents, what we do is we just take one step after another. And we realized there's no way we could do this alone, so we began reaching out to the broader CDG community, but we also knew we needed to have more NGLY1 patients. So I'll skip over the details of how we did this. There's an article in the New Yorker explaining what we did, but the bottom line is we used the internet, some social media, a little smarts about Google to find other patients. And find other patients we did, and rather quickly as it turns out. So within just a couple months, we actually had 2 more NGLY1 patients in Israel, in Turkey. After that, and then a few months later, we had 2 more in Israel. I think a week or 2 after that, we had one in Palo Alto. And over the past 3 years, NGLY1 patients have been popping up all over the world for a brand new disease. And what this has done is it has really turned that darkness of isolation, that time on undiagnosed island, and washed it away with the sunshine of community and science. And that community, I can guarantee you, is very united. In fact, there's 3 NGLY1 families here today, three of the families that are out there. And it is a sign of solidarity that we're all working together. And in fact, after last week, in which case where another case turned up, we're now 38 strong. So in the past 3 years, we found 38 cases of NGLY1 around the world, up from 0. And once you have a community, you can start having meetings. So we've probably had about 4 patient-scientist meetings so far. This is the first one back in 2014. And as soon as you have a patient community, you can create nonprofit foundations, as others have done. So in fact, there's not 1, but 2 nonprofit foundations for NGLY1 now. I'm the president of NGLY1.org, and my fellow parent and friend Matt Wilsey leads the Grace Wilsey Foundation. And we've also begun realizing that, well, we've got to do some research, and research takes money, so we've been on a fundraising campaign as well. So NGLY1.org has raised and invested over $500,000 for NGLY1 research in the past 3 years, and the Grace Wilsey Foundation has literally raised millions. So we've been very aggressive in going after the science behind this disorder. We've participated aggressively in the natural history protocol at the NIH for NGLY1 or for CDGs, and I would strongly encourage anybody who hasn't done that to do it. It's a fantastic experience. So we're very grateful to Bill Gall for enabling this to happen, but especially to Lynn Wolfe, who I sense really runs the show here and really makes this actually happen. And actually, if for no other reason, you should go just to watch Lynn Wolfe in action, who is literally 3 parts human being. She's equal parts clinician, scientist, and field marshal. It is truly a sight to behold. So if you haven't been through, what I've got here is a single picture for every poke, prod, procedure, or appointment that occurs during the course of this week. So, you saw the spreadsheet schedule earlier from Lynn. That's what it actually looks like when it happens. It's an intense week. So what happens when you get a large number of patients through a protocol like this, and so far I think 13 NGLY1 patients have been through, are mountains of data. And with mountains of data on lots of patients, you actually start to get statistical confidence in your results. And in this case, you're starting to get biomarkers, clear indicators of what's happening inside the disorder. And these are actually quite critical. If you want to go to somebody like the FDA and say, we'd like to do a clinical trial, they want to know what your biomarkers are. So that's why it was one of the many reasons why it's critical to participate in something like the NGLY1 natural history, or the CDG natural history study. And in the case of NGLY1, we're even finding some very surprising things, like hypoglycosylation, which is not entirely what you'd expect at first hand from a disorder primarily involved in deglycosylation. This is something you'd expect to see in a more classic CDG. We've also been busy with HUD developing assays, and assays are important for measuring the activity of an enzyme, in this case, the missing enzyme NGLY1. And once you have a robust assay, you can proceed to high-throughput drug screening, where you could have machines actually test thousands of compounds against the assay to see if any of them can potentially restore the activity of what's been lost. So assays are a critical first step. The community has been investing in the creation of model organisms. We have, to date, mice, flies, zebrafish, worms, and even yeast. Now, I don't know if ours have the same wonderful nutty flavor as those pictured here, but we certainly have them by now. Yes? Wow, so we have 8 mice.
8 mice
8 very brave mice who will give their lives for NGLY1. We've also, I'm sure like many of you, been pursuing enzyme replacement. You know, if you're missing an enzyme, a natural strategy might be to replace that enzyme. And one interesting thing about NGLY1 is if you're trying to synthesize it and get it back into cells, that at least to some degree that first step seems to be done if you have access to the internet. So if you do a little Googling on synthetic NGLY1 proteins, what you'll find is that it's already available online, except that it costs $402 for 10 micrograms. Which if you work out roughly therapeutically what that would be, even if it were to work, it's around $2 million a week. So you may have to talk to your insurance company about whether or not a strategy like this is appropriate. But you know, at least somebody has taken that first step. Now it turns out there's other issues with actually getting the enzyme to the right parts of the cell, but you know, those are scientific obstacles that people in the community are actually looking at very actively. We've also been engaged in some crowd science that's relevant not only to NGLY1, but to all of CDGs, because we've been partnered with the Sioux Lab at Scripps to do a project called Mark to Cure. What Mark to Cure is doing is it is systematically allowing volunteers to biocure. It could be, by volunteers, I mean, it could be the people in this room. You don't have to be a scientist to do this. You can go to marktocure.org, and you can start marking up abstracts in PubMed related to CDGs. And what we're doing is we're curating this knowledge in a way that makes it systematically mineable and connectable, so that we can actually look for connections between CDGs and connections between CDGs and potential therapies. So this is definitely an experiment, but so far it's going quite well. And again, anybody in this room can participate in this project. And if you do participate, I guarantee you will learn a lot more about CDGs than you ever thought possible, because you'll be looking at a lot of abstracts. There's been a lot of basic science going on in Hud Freeze's lab and many others regarding NGLY1. And some of these findings have actually been enabling of therapeutic opportunities. In particular, there's suggestions in the literature now that allow us to believe that NGAZ inhibition is a reasonable therapeutic route for NGLY1. In fact, it's so credible that the pharmaceutical firm Retrophin has said, you know what, we're willing to create a drug discovery unit around this approach. So they're actually going to start looking for NGAZ inhibitors to see whether or not we can actually bring this to the patients. And I myself have shifted much of my research in computer science into really the intersection of computer science, biology, and medicine. Just because I feel like I owe my time to my son and to the entire community. And I recently had a grant proposal funded on developing a drug development platform for NGLY1 so that we can start screening some of the suggestions that are out there. It's a very simple platform, as it turns out. We're using something called planarian worms. And the nice thing about these guys is once you've got them created, if you want more of them, you just cut them in half. And if you cut a planarian worm in half, you have 2 planarian worms. So you can grow them up quite quickly once you've got them. And what these brave worms will be doing is testing out around 40 different therapeutic predictions that have been made for NGLY1. So if the worms get better, for some definition of better, then maybe humans would too for that particular drug. We've been quite heavily engaged in advocacy and outreach because it's not all about science. There's actually a lot of regulatory and policy obstacles in our way too. And that's really not in just, that's in more than just our way, it's in everybody's way, and really everybody in rare diseases way, I should say. So we've been very active in trying to clear those. So for outreach, we created a website. We got an NGLY1 gubernatorial declaration for an NGLY1 day. My wife co-organized a rare disease conference in Utah to spread the word. Held a rally at the state capitol for a new precision medicine clinic for rare and undiagnosed disorders at the University of Utah. An initiative was actually been funded and supported. So at our local hospital, we'll have a clinic we can go to, or anybody can go to with a rare and undiagnosed order for very precise treatment. We've been attending academic conferences looking for other patients and interacting with scientists. So we attended ACMG, and in fact, we set up a booth at ACMG. Actually, I should say my wife set it up. She did all of that, actually. And then manned the booth interacting with clinicians saying, this is what an NGLY1 patient looks like. And in one day, found 5 more NGLY1 patients. Just from talking to clinicians who said, oh yeah, I diagnosed one of those. And we're like, well, why didn't you connect them to the community? They said, well, there's no cure. So that's what's happening out there. So if you're a clinician and you diagnose a disorder, please Google it. And if there's a community, connect them up. And again, in terms of regulatory and legal obstacles, my wife actually went and testified to the State House and State Senate in Utah to get a law passed called Right to Try. And what this does is it gives anybody in Utah who has a terminal condition the right to try any medication past phase 1 FDA safety testing, which, believe it or not, had actually failed the prior year. So that's now law as of July 1st in the state of Utah. And I myself have been engaged quite heavily over the past 9 months with some folks from the White House as part of the Precision Medicine Initiative, which is something the President announced back in January. And what this is all about really is about implementing the funding, the policies, and the initiatives necessary to bring genomic science and medicine to the entire population so that everybody with a rare disease can benefit from these advances. And so that really there's a sense of hope for all of us, I think, if we can get this initiative through and actually get it up and running. So my parting thought in terms of action is that the phrase not actionable has long since outlived its usefulness, and it's time to get rid of it, because in the absence of any available action, it is always possible to do science. Thank you.